Good morning. Today we're talking about protein synthesis. That's taking DNA and building protein traits for you inside of your body. It sounds like a small process, but it's actually taking DNA and building you with all of the protein traits that make up who you are. Protein synthesis has a few different steps. One of them is called transcription. And transcription, we're taking DNA and building a messenger RNA. And the second step is called translation, where we take that messenger RNA and we build an amino acid. Now, to start us off today, I'm going to tell you an analogy, how I view this process to just keep it in perspective. So up here, this top statement is called the central dogma of protein synthesis. This is where we take DNA, we build an mRNA strand, and then from that mRNA, we build a protein trait. This is much like, so for my analogy, DNA is much like a cookbook with thousands and thousands of recipes inside of it. And the first step, we take that cookbook and we copy down one single recipe. Like if we want to make bread, we would copy down the single recipe for making bread. That is like our mRNA strand, that single recipe or that single gene that we want to then make a protein from. The second step is going to take that single gene and that single recipe and then make your bread or make your protein trait. So hopefully this helps you keep in mind what this big process is like inside of your body. Now, just to review the players, DNA, remember, is a double helix. It has deoxyribose and it has this thiamine group in it. It will be required for protein synthesis. And there's also RNA. So we haven't talked tons about RNA since our first lecture. And RNA is single-stranded. It has a ribose and a different nitrogen base. Instead of thiamine, it has uracil. But we need all three kinds of RNA to complete protein synthesis. So the three kinds are messenger RNA. That's like our recipe that's been copied in that first step. We need ribosomal RNA, which is rRNA, and transfer RNA, which is tRNA. Down here in this picture, you see all three kinds of RNA. You see down here in red, the messenger RNA is a single copy of a single gene that we want to make a protein from. Then this brown piece here is almost like a Pac-Man, and that Pac-Man is the ribosome. It has this lower kind of small lip and then a bigger cap. When the messenger RNA gene comes along, then it's going to bind to that lower lip, and then the whole lid, the large lid, falls in place, and this particular ribosome is going to ratchet down our gene from a starting position to a stopping position in order to make a protein. The last RNA are transfer RNAs, and they come and they bind, delivering an amino acid up here in purple to our code. They deliver the amino acid that then grows into a polypeptide or a protein. And those transfer RNAs allow us then to build this protein. So we need DNA, but we need all three kinds of RNA as well. Now, where did we get these names, transcription and translation? So a scribe back in the olden days was someone who copied text, right? So they would make a copy of a text. It didn't necessarily need to be in a new language. It was just a word-for-word copy kind of before printing presses. And so our word transcription has that word scribe as a part of its base. And so this first step, we're going to take DNA and we're going to make a copy of a single gene. And that's exciting because that allows us to copy that gene that we want to then turn into a protein trait for us. In transcription, we do not change chemical languages. Look at up here, we start with nucleic acids and we end with nucleic acids. So we're not changing languages. And this has to occur in the nucleus because the DNA is too big to get out of the nucleus and go roaming the cell, right? So this part of the process will occur in the nucleus. The second step is translation. And in translation, this is like a UN translator that sits and translates, German to English or French to German. And translators translate between languages. And that's what's happening in protein synthesis for this second step. We're going from nucleic acid language, that's a certain kind of macromolecule, to a different kind of chemical language, amino acid language. And so we are translating into a new language. This particular step happens at the ribosome, and ribosomes are either free inside of the cell or located on the rough ER. So this particular step has to happen in the cytoplasm at a ribosome, so outside of the nucleus. Now, let's go through an overview of transcription. There are actually 4 different parts that I want to talk about today. There is transcription, and then something called mRNA processing, then translation, and then something called protein processing. So I want to walk you through each of these steps. Just to give you an overview of transcription, transcription occurs when your body gets signals from other places. Those signals are often known as transcription factors, and they're going to flow from your brain and other places in your body to the genes that can be transcribed or copied. So in the case of insulin, you might be thinking about food, you might have hunger signals coming from your digestive tract that will release these transcription factors that will then flow through your body to your pancreas, telling your pancreas to make some insulin. It will say like, hey, there's food coming, so make me some insulin, right? Make me this particular protein product. These transcription factors are going to bind at the starting position of like sort of DNA, at the beginning part of the gene for insulin. Every gene has a particular starting position and kind of a stopping position as well. These genes then are read only in one direction, just like a sentence has a capital letter and a period telling us where to begin reading and then where to stop reading. So all these transcription factors come from other places. They're often signals. They'll bind at that starting position. Once that those transcription factors bind, it creates some stickiness on the code right at the beginning part of that gene. This enables RNA polymerase to bind. And once RNA polymerase, that enzyme binds, it will then read the gene from sort of that starting position all the way to the stopping position. We actually build our new mRNA code, right, our copy of our gene. We build it in the 5' to 3' direction. So RNA polymerase builds in that 5' to 3' direction. And it's going to build a complementary strand of DNA. So if you look over here, if DNA reads AAA and we're building an RNA complement to that, then rather than having TTT, you would have UUU for your mRNA. And so your sort of RNA polymerase will then read that gene all the way, copying it complementary to that original strand all the way until that stopping position. This occurs in the nucleus. Now, our mRNA has been copied and it will be released from the cell. Once it reaches that stopping position, RNA polymerase falls off. The mRNA strand, the copy, is then free inside of the nucleus. But we can't just send it out into the world, right? Without protection, without kind of processing it a little bit and making it ready to do the third step, which is translation, or that second big step called translation. So we process mRNA. mRNA is processed in several different ways, but over here, if you look at my picture, one of the things that happens is all these introns get taken out. So they get cut out of our mRNA. Introns can be there for spacing. They can be there to signal kind of positioning, like where the beginning of a gene is and where the end of a gene is. So they usually have other sort of reasons for being a part of that original mRNA piece. But they're not necessary to actually read the gene and make the gene. So we can cut out those particular introns. We splice together. So we like sort of continuously splice together the exons. I remember exons get to exit and go out of the nucleus. And so the exons get spliced together, these darker red regions, to make what part of the gene is important that you're going to then read to make a protein trait. The last thing that we do in mRNA processing is we add a 5' G cap. That's a guanine cap, a whole bunch of guanines sort of on top of at the beginning or the starting position of that gene. And then we add a poly A tail, a whole bunch of adenines at the end of our transcript or our mRNA gene that we have just copied. This is kind of like if I were to send you out into the world, I wouldn't send you out without a hat and a pair of shoes in the winter, right? So this is a way to put like a hat and a pair of shoes on your mRNA transcript so it's ready to like sort of go hiking around the world, right? In this case, it's not going to hike out into the world. It's actually going to go to a ribosome. So let's talk about the second big process or part of protein synthesis. It's called translation. And in translation, what we're going to do is take our copy of our mRNA code, and then we are going to use it to produce a protein, some kind of protein trait. In the case of insulin, we would use the insulin gene to then produce insulin that can be released to your blood and then regulate your blood sugar. This process is then going to convert, up here in red, this mRNA strand into a new language, into amino acid sort of sequence, or a polypeptide, or a protein. Now, interestingly enough, in mRNA, every 3 letters of mRNA represent something called a codon. And these codons, each 3 letters or each codon, will represent one specific amino acid. There are more than 20 amino acids in life or in living things, but there are 20 very, very common ones that we see and we code for in DNA. Remember, translation is going to occur in the cytoplasm at a ribosome. So how does that work? Your mRNA has just been given a G-cap and a poly A tail, and it has its exons. As it exits the nucleus, it's going to find a ribosome, one of those like kind of Pac-Man shaped rRNA pieces. When it binds, the starting position will attach to the lower lid of the Pac-Man. When that sort of starts, then the start codon, the first 3 letters that represent start, get to bind at that starting position. Once the start codon binds, the rest of the Pac-Man sort of falls in place. Then this is actually going to move down your sequence every 3 letters, and it's going to bring another transfer RNA. Those transfer RNAs will dock, deliver an amino acid, and then as we read every 3 letters, you're going to build your protein by adding more and more amino acids. This will continue until your sort of rRNA reaches the stop codon, in which case a release factor is bound, and the release factor makes the rRNA release the transcript. The transcript is then free. It can bind again and be read again if you need more protein product. It can also be degraded inside of your cells if you are done making that particular protein product. So you're going to read every 3 letters down your mRNA until you reach that stopping position. And then you've produced a sort of a sequence of amino acids. Now, there are 20 amino acids that are really important for life. Here is a codon table. It's interesting that the language for these codons is universal. So if you have UUU, then that represents phenylalanine, that particular amino acids. Or if you have CCU, then that represents proline. Notice there is only one start codon, AUG, and there are 3 possibilities for that release factor or that stop codon in our language. And all these other ones are the main sort of amino acids that we see quite commonly used in life. Now, codons, we said, are 3 letters of mRNA code that represent one protein. The anticodon then is carried on the tRNA. So look down here at this picture. In red, you have mRNA. So every time you have UUU, then that's the codon in red. The anticodon is the complement, AAA, and that's delivered by the tRNA. A tRNA has an anticodon on one end, and then it delivers a specific amino acid. So UUU, for that codon, always means phenylalanine, that particular amino acid. So this is why your cells, or how your cells, can deliver a very specific sequence of amino acids in order to build a very specific protein for you. Now, once you're done building those proteins, then sometimes the proteins are processed. Again, we don't just turn the proteins loose, right? Sometimes proteins are in an off position, so we have to turn them on. So sometimes different chemicals will be added. They will be folded into new shapes so that they're in working condition. Sometimes we send proteins to the Golgi apparatus so that they can be further packaged. Sometimes proteins need external pieces like cofactors and coenzymes that are added, like metals and things like that are added to them so that they are working. Usually those are things that you're eating in your diet that then are placed into the protein as they are processed. But proteins are processed and then often go to the Golgi. They are then marked and shipped out of the cell or shipped within the cell to the locations where they are required. So this gives you an overview of protein synthesis. Again, this is taking DNA and building a protein trait for you. So it's really the way that we take our DNA code and build you. So have a great day, and I will see you next time.